Hello class, my name is Franzia Desgranges and I'm a biology major, 4th year, and today I'm doing my presentation on bacteria DNA sequencing. The bacteria that I'm doing my presentation on is Pseudomonas aeruginosa. So here guys, this is just an image of the DNA sequence that Dr. Long has sent me, and when I pasted this into the DNA sequence match, this is what I got, which was the bacteria, which is the Pseudomonas aeruginosa. So I'm going to go a little bit in depth about this bacteria. So Pseudomonas aeruginosa is a gram-negative bacterium, which is an opportunistic pathogen that is normally in habitats such as soil, surfaces, and in aqueous environments. And it also exists in diseases or conditions such as cystic fibrosis and traumatic burns. Pseudomonas aeruginosa is the second most common organism isolated in nosocomial pneumonia. And it's also the third most common in both urinary tract infection and surgical site infection. Pseudomonas aeruginosa is also a common cause of nosocomial infection that's usually found in hospitals, and it usually can be transmitted due to patient contact. Like if a nurse can give a patient this type of bacteria. So it's mostly found in hospitalized area environments. So I'm gonna go a little bit in depth about this bacteria. So Pseudomonas aeruginosa is a gram-negative bacterium, which is an opportunistic pathogen that is normally in habitats such as soil, surfaces, and in aqueous environments. And it also exists in diseases or conditions such as cystic fibrosis and traumatic burns. Pseudomonas aeruginosa is the second most common organism isolated in nosocomial pneumonia. And it's also the third most common in both urinary tract infection and surgical site infection. Pseudomonas aeruginosa is also a common cause of nosocomial infection that's usually found in hospitals, and it usually can be transmitted due to patient contact. Like if a nurse can give a patient this type of bacteria. So it's mostly found in hospitalized area environments. In this slide are some images of the Pseudomonas aeruginosa, and on the right is just an image of the rod-like gram-negative pinkish color type that you'll find in an organism, like if you're doing the microbiology aspect of it. And in the left hand, that's what you'll find as far as like in the slides. If you are doing a slide, you'll find that on the left side of it. So that's basically a brief or image of how it would look like of the bacteria. So in this slide, is more of the microbiology aspect of it. This is just an image of a petri dish, which is the four-way streak of the Pseudomonas aeruginosa. And Pseudomonas aeruginosa is also an aerobic bacteria, which is necessary for optimal growth. And most strains will multiply slowly in an anaerobic environment. And Pseudomonas is preliminarily identified by typical odor in vitro. And as we talked earlier, Pseudomonas is a gram-negative rod shaped, which is usually commonly found in free living and moist environments, but it's also a pathogen of plants, animals, and humans. And in this slide, these are the infections that Pseudomonas aeruginosa would cause. Some of the infections that are caused by Pseudomonas aeruginosa are urinary tract infections, respiratory system infections, dermatitis, soft tissue infections, bone and joint infections. Also, gastrointestinal infections and a variety of systematic infections, which are particularly in patients with severe burns. And here in the left, it's just images of the infection on the foot, and this is what it will cause as far as in severe burns and in the patients, and also in the right, it's also in the arm that can also cause that too as well. And in this slide, chronic infections can lead to progressive disease and cystic fibrosis, which is regularly complicated by antimicrobial resistance. And in this slide, it's just basically treatments, drugs that are used to treat this bacteria. And some drugs that are used to treat this bacteria are aminoglycosides, carbapenems, cephalosporins, which is the third generation of that drug, and the fourth generation of this drug is cephalosporins, which are all drugs that and antibiotics that are used to treat this type of bacteria. And here's the hierarchy view of the matches that I found. And for the domain is phylum, class, order, family, and genus. And for the bacteria under phylum is Proteobacteria. For the class is Grammateolobacteria. The order is Pseudomonas. The family is Pseudomonas Daxia, and the genus is Pseudomonas. And in this slide, just more of the matches that I found after putting in the sequence match after I put my DNA sequence in there. And then this is just more basically going in depth on the matches that I found. And this slide is just the Bayesian classifier. And that's when I put the sequence DNA match into that classifier, and then it gave me the result, which was 100%. So that's the just a basically brief description of that. Is just the questions and results, and the question is, do the sequence match and the Bayesian classifier assignments match? Why might they differ? So in the previous slides I had shown you the result or the description of the Bayesian classifier, and I definitely got a result which was 100%. So yeah, the sequence match and the Bayesian classifier assignment showed the same result from my DNA sequence. Both of the results for the database proved that it had the same hierarchy. And the second question is, based on the taxonomic assignments, what can you infer about your unknown in terms of ecological importance, usefulness to humans, and potential to cause disease? What this type of bacteria can spread in hospitals via the hands of healthcare workers or by hospitals' equipment that is not properly cleaned. So Pseudomonas infections are considered opportunistic infections. This means that the organism can only cause disease when a person's immune system is already impaired. So basically, it's just a more usually found in hospitalized areas. And treatments and antibiotics have been found that are useful to humans to treat this type of bacteria. And this slide is just a description of the blast DNA that I received for my bacteria of the DNA sequence, which is Pseudomonas aeruginosa. And this is just basically a short description of how it looks. And this slide is just another description image of the taxonomy report of Pseudomonas aeruginosa. And in this slide is just a brief description, the breakdown of the phylogenetic tree of the bacteria of Pseudomonas aeruginosa. And in this slide, I basically just had a brief discussion of the pathogenicity and the epidemiology of Pseudomonas aeruginosa. And the pathogenicity of Pseudomonas aeruginosa have several virulence factors, but their roles in pathogenesis are unclear. All strains have endotoxins, which is a major virulence factor in bacteremia and septic shock. And in the epidemiology of hospital infection due to Pseudomonas aeruginosa is a primarily non-zoonotic pathogen. Pseudomonas aeruginosa finds numerous reservoirs, disinfectants, respiratory equipment, food sinks, taps, and mops. And these organisms is often reintroduced into the hospital environment on fruits, plants, vegetables, as well by visitors and patients, transferred from other faculties. Spread occurs from patient to patient on the hands of hospital personnel by direct patient contact with contaminated reservoirs and by the contamination foods and water.